Hi there, and welcome to this video on GCSE Biology for the AQA Specification, focusing on the topic of organisation of an ecosystem, and in particular on the levels of organisation. I'm Shumina from StudyMind, where we help you revise GCSE Biology with our helpful video tutorials, tailored to your subject, your specification, and to you. If you're new here, make sure you click the subscribe button. Whilst you are watching, please leave any comments below if you're unsure about anything, and let us know if it's your first time watching our videos so we can send you our free revision materials. We also have helpful timestamps below for each part of the video to help guide you through the specification. So let's get started. Hello there, and welcome to this tutorial on levels of organisation. So in today's tutorial, we are going to do a little recap of levels of organisation. We've already visited this in a previous tutorial. We're then going to have a look at how we can present data, so looking at kite diagrams and also averages. So these are our specification points today, so have a read through them now, perhaps pause the video at this point, have a read through them, and come back to them at the end and just make sure you understand everything. So let's get on with it. So biomass is a term we've already come across in a previous tutorial, but just to recap, it's used to describe getting energy from biological matter. So producers photosynthesize in order to produce biomass. They are fed upon by primary consumers. Now remember, producers are usually a green plant or an alga, and they make glucose from photosynthesis to provide energy. Now, food webs can track the feeding relationships of organisms. So let's have a look. So we have our producer here, which is our grass, so we label that up first. Next, we must label the primary consumers in the food web. So these are the organisms that consume the producer. So we have rabbits and mice here. Next, we label the secondary consumers in the food web, so the snake. And next, the tertiary consumers in the food web, so the owl and the hawk. And this is the final product, a fully constructed food web. Now, sampling is a technique used to find a small proportion of a population and use it to draw conclusions regarding the whole population. So this is a definition you need to learn. And how can we sample? Well, there's many different methods, but in your specification, it asks you to know about transects and quadrats, which are measurement tools that can be used for sampling. So quadrats are just simply these structures over here, so often made from wood or wire, that form a 0.25 metre squared area. And you can place a quadrat over an area and use it to sample. So this can be done by either counting the number of a certain organism, or finding the percentage cover of an organism or the area within the quadrat covered by the organism, or observing the diversity of species in the area. So we do that by just observing within this area that the quadrat covers. So for example here, if we were to place a quadrat over a woodland area, for example, then we can either count the frequency of the species of trees, we can say there's 13, but that doesn't actually tell us that much, so perhaps we might want to think about the percentage cover, for example. So we can say more than 50%. Now that's just by using our eye and saying, okay, well these trees are covering around 50% of the quadrat. Or we can look at species diversity, but here we can see that they're all one particular type of species. So it's really important that we make sure our sampling is random in order to produce valid results and ensure no bias. So this can be done by throwing the quadrat over an area with eyes closed to avoid bias, but an even better method involves two measuring tapes. So what we do is we place two measuring tapes across an area like two axes of a graph, and then we can use a telephone number, or even better, a random number generator to make random coordinates. Then you place the quadrats at those coordinates and repeat this at least 3 times for reliability. So there's just a few things I want to make clear to you from this slide. So if they ask you in the exam how you might generate a method for random sampling, then you could say place two measuring tapes across an area like two axes of a graph, and then use a random number generator. So that was always my go-to answer in exams, so using a random number generator to make random coordinates. And another thing I want to point out to you is that we need to repeat. So anytime you're describing any sort of experiment, make sure you repeat it, because we need to do this at least 3 times for reliability of our result. The more times we repeat, the more reliable our result. So, for example, here, we might lay out tape like the axes of a graph, so kind of like, oopsie, I'm really bad at drawing straight lines, but let's go for it. So, kind of like an x and y axis of a graph. And then let's say your random number generator comes up with a 50 meters along the x and a 100 meters along the y, then that would be where you placed your quadrat, for example. So, belt transects can be used to sample changes in diversity across a range of areas. So you can use them to monitor species diversity along a rocky shoreline, because you're placing the quadrat at different distances along this line, which can be built using measuring tape. So you'd have your measuring tape going across like this, for example. These are your different points. And you'd place your quadrat at different points along here. I'll just draw a box just to give you an idea, and you're sampling at each of these points, okay? So that means that you can monitor species diversity, because here there might be a higher percentage of dandelions, but here there might be a higher percentage of oak trees, for example. So we're placing the quadrat at multiple points along the measuring tape. So here we are, here is an example drawn out a little more clearly for you. So we might be sampling rocks, it seems, in this picture. So we have our measuring tape, we have our quadrat, we're placing at multiple points along the tape, and we can see that we can then monitor the distribution or the density of whatever we're sampling as we go further along the measuring tape. So you can use a transect to see the effect of different abiotic and biotic factors on species diversity along a habitat. You can then use this information to draw a kite diagrams. This allows us to present our data. Also, just a little point here, make sure you can understand how to extrapolate information based on the proportions found using a quadrat. So for example, if you have a 1 meter squared quadrat and you count 6 buttercups, then if you were to have a 3 meter square field, you would assume that there might be 18 buttercups, so 6 multiplied by 3 equals 18. So that's what we mean by extrapolating information, applying what we learned from the quadrat to reality and using it as an estimate. So this is a kite diagram. So kite diagrams are used to measure the number or percentage of something against distance along a transect. So you can see here on the x kind of axis it's distance, and along the y-axis it's the type of organism that we're sampling. So all it's showing is the number or percentage of something against distance along a transect. So the axes are plotted as shown, and the distances of the transects are plotted on either side of a horizontal line representing the species. The eventual result will be points on either side of the lines. Once joined up and coloured in, they resemble the kite shapes seen here. So if we look at this data, we can analyse the species diversity across the transect. So this data was taken from along a habitat. We use the width of the bar from the midline for each organism to see how many individuals are present. So for example, with the tree and shrubs, so we go from the midline and we measure either side and that's an equal proportion to the number of trees and shrubs present. So therefore we can see here that at the first sight, lithics were the most abundant organism. However, as we went further along the transect, other organisms such as trees and shrubs became more and more common, whilst the lithics reduced in number. And at the final site, we see that reeds and rushes, bush rushes, were the most abundant. Yes, so as we can see, reeds and rushes become more abundant down here, but we can see initially at the start of the site, so at the very beginning of the measuring tape, we have lithics are more abundant. So at the first site, lithics were the most abundant organism. However, as we go further along the transect, other organisms such as trees and shrubs become more and more common, whilst the lithics reduced in number, and we can see that the lithics reduced in number. Sorry if I just go back, because you can see that they're trailing off around here, whereas the trees and shrubs, the distance from the midline to the point is getting larger. And at the final site, we see that reeds and rushes were the most abundant. As we can see here, they stay pretty low density up to site 11, and they start to increase in abundance. So that is interpreting kite diagrams. So you can see it's pretty simple. And if you understood when we went through it just now, then you'll be able to apply that exactly as we just did it to the exam. So just a few things for you to remember. Your site is along the x-axis, or your distance is along the x-axis. Your organisms are along the y, and it's just measuring the distribution or density of species. So we measure from the midline to the point, and that gives us an idea of how densely populated with this species the area is. Now let's have a look at some mathsy stuff. So averages. So I'm sure you've done this many times, but let's recap. So the mean, if we have many sets of data, we can attain one average value by a calculation of the mean. So for example, if you have taken data from 3 randomly placed quadrats, regarding the number of lichens, you can work out the average number. So in order to calculate the mean, you have to add the values and divide by the number of values. So you can see here for this organism, we've added the values. Here we've got 41, and then we've divided by the number of values. We can see that we have 3 values because we have 3 quadrats, and that gives us 13.67. We're then going to round up to a whole number because we're talking about organisms, so that's really important to do. So we're going to round up to 14. And that gives us the average number of the organism within the area. So in order to calculate the median, which is a different type of average, you place all of the numbers in ascending order, and it's really simple. The number in the middle is the median. So for example, if you have 9 quadrats and have counted the number of lichens, you get the following data: 1, 1, 3, 4, 5, 6, 7, 7, 7. We've put it in order. All we do is we hop from number to number until we find the middle number. So what I like to do is get my finger and hop on each side. So I would be doing this with my fingers rather than with a pen. And then I reach the middle number. So that's 5, so that's our median. So, but it's really important that you put the numbers in order. So remember to order the numbers first in ascending order and then find the median. And just in case, I just remembered, if you have 2 numbers as the middle number, so let's say I added an extra number over here. That would mean that we would then have an even number, and therefore we would have 2 middle numbers. So I'll just show you now. So our 2 middle numbers would be 5 and 6. Now, how would we find the median of that if we've got 2 middle numbers? Well, then all we would do is find the average of those 2 middle numbers. So we would do 5 plus 6 over 2, which is 5.5. So that's just showing you what to do if you have 2 numbers in the middle rather than just the 1 number as we had in the original scenario. So all you would do is just take the average of those 2 numbers, the mean of those 2 numbers, as we did on the last slide. So 5 plus 6 divided by the number of numbers, which is 2, equals 5.5. Now the mode is the most common number. So if we use the same data as before, the mode would be 7, as it is the value that we see the most. And you can draw bar charts in order to plot such data. So you can see how this information can be presented in a number of different ways. Okay, so our final specification point. So in previous tutorials we saw that producers are eaten by primary consumers, which are in turn eaten by secondary consumers and then tertiary consumers. Now predators kill and eat other organisms, so the predator of the producer is the primary consumer, and the predator of primary consumer is the secondary consumer. So whatever eats the other is the predator, whereas prey are killed and eaten by predators. Therefore, producers are the prey of primary consumers, and primary consumers are the prey of secondary consumers because they're being eaten by the secondary consumers. Now there is a predator-prey cycle. So for example, the lynx and the snowshoe hare follow this pattern. So they fluctuate with one another, the predator and the prey. So as the predators increase in number, the prey will reduce because they're going to eat the prey, leading to a reduction in the predators, because then there will be less prey for the predators to eat, so there will be less food, so the number of predators will drop. So I'll just explain that a little bit slower. So when our numbers of hare are higher, our numbers of lynx are lower. Then as they start to come down, that's because the lynx are eating the hare, so we get a rise in the number of hare. And then as that occurs, we get a drop in the lynx. And then when the hare population is high again, the lynx have more prey, so it's going to rise, and so on. So it's always a cycle, because as the number of hare reduce, as the number of lynx increase, the number of hare reduce, and vice versa, because as the predators go up, the prey will reduce. So this is known as the predator-prey cycle. So just showing it to you in a little bit of a bigger format. So as the lynx population decreases, the hares can survive longer, so their population increases. So you can see as the lynx population decreases, the hares increase. So that's seen in all of these patterns here. This then provides increased food for the lynx in turn, so then the lynx population will increase, and the lynx will eat the hare, then causing a drop in the hare population as the number of predators increases. And we go on like that. So that's our predator-prey cycle. Okay, so we've covered all of the specification points. Let's have a look at a practice question. So I'll read through the statements, you can then pause the video, have a go, mark which ones are true and which ones are false and why, and then we'll go through the answers. So A, as the population of hare increases, the population of lynx decreases. B, producers have no prey. C, the mode is the middle number in the data set. And D, belt transects can be used to carry out systematic sampling. So let's go through this. So A, oopsie, it's changed. So A should say, as the population of hares increases, the population of lynx decreases. That's not quite the right statement there, my apologies. But that is true because of the predator-prey dynamics. Then B, producers have no prey. That is true. Remember, producers are right at the bottom of the food chain, so they have no prey. C, the mode is the middle number in a data set. That is false, that's our median. And D, belt transects can be used to carry out systematic sampling. Also true. So well done for today. These are our specification points covered, and I will see you for the next tutorial. Thanks for watching this free video from StudyMind. If you liked this video, make sure to subscribe to catch our newest videos by clicking below, and leave a comment on a topic you'd like a video on. Click here to watch more videos in our series for GCSE Biology, or visit our website studymind.co.uk for free past paper compilations by topic and specification.